Protein rating! Sometimes one protein is actually a bunch of proteins in one complex, and that's the case with the subject of today's rating, brought to my attention by this lovely person. POLD1 or P125 is shown here in green, and it is part of the catalytic subunit of DNA polymerase delta. It holds the DNA and helps to replicate the lagging strand. This is important because otherwise polymerases would look at the 5' to 3' strand and be like, what do I do? The two strands of DNA are different from each other because of
chemistry.
So, one of the strands is a cakewalk to replicate. That's the leading strand, the 3' to 5' strand. POLD1 deals with the strand that's harder to add bases to because of
chemistry.
By adding fragments of DNA at a time. This requires an RNA primer in order to overcome the chemistry that prevents the addition of bases to the 5' end. POLD1 uses the primer as a marker to tell where it needs to make its replications, and then it yanks it out so that eventually there's only DNA left in the lagging strand, and thus our genomes are replicated successfully. POLD1 is also a proofreader, aiding in many different DNA repair pathways. Mutations in POLD1 or in any of the proteins associated with the DNA repair pathways can lead to accelerated aging, as in Werner's syndrome, or genomic instability, which is associated with cancer. 10 out of 10 allows us to replicate both halves of our genome despite the challenging
chemistry.